Good morning. My name is Dr. Dick Montali. I'm head of the Department of Pathology at the National Zoological Park, Smithsonian Institution in Washington, D.C. My title is Dynamics of Disease Transmission Between Wild and Captive Population of Animals. What I'd like to do during this hour is to describe, using my experience with disease problems at the National Zoo and in some other settings, wildlife, the transmission of diseases from the wild to zoo or captive animals that are in captivity. In addition, talk about the reverse, the transmission from the captive to the wild populations, and in addition include some discussions about similar dynamics between domestic populations and natural animals in natural habitats. What I'd like to do is to first begin with some concepts of the types of diseases that we see, and we'll refer to those diseases in wild populations as wild diseases, and those in settings where animals are held in captivity as captive diseases. For example, diseases that I can use as a good example would be rabies, which is a disease that often occurs in wild populations, has a very wide host range, and can enter captive settings, and I'll show examples of this. Diseases that might move from natural habitats into domestic populations, on the other hand, would be a good example of this would be bovine tuberculosis via the wild animals that actually have been known to carry this disease. Badgers in the UK and brush-tailed opossums in New Zealand actually have been responsible for carrying Mycobacterium bovis into the domestic cattle populations. Well, my orientation mainly is working with captive diseases, that is, diseases in a large zoo setting containing numerous taxa of animals. And I've outlined here basically the main sources of diseases that get from the outside to the inside of a zoo. And many of the diseases arise within the colonies of animals themselves via the various different pathogens, viral, bacterial, fungal, are diseases that are inherent within the colony. In some cases, if these diseases have a wider host range than that particular species, then they can cross over into other colonies. One of the situations we deal very commonly with are diseases that are transmitted from the wildlife in the area. For example, there are many raccoons, rodents, and roaches, as I have noted as the 3 R's, are often responsible for many of the viral, bacterial, and parasitic diseases we see in our captive species. And I will illustrate some of these as we go along. There are other wildlife, fox, skunk, some of the small rodents that live freely in zoos, and also feral animals like cats. Cats actually often harbor in zoo settings and carry diseases like toxoplasmosis, of which there are a number of susceptible species for this disease, mainly the marsupials. And then there are food sources. Certainly there's a number of ways diseases can get into a zoo setting through food sources. Domestic pets to some degree, but less so these days. and of course humans, mainly with related species between great apes and humans. There are zoonotic diseases that go both ways, so these are the main sources of diseases that often represent many of the infectious diseases we struggle with in zoo situations. I mentioned that I've divided the diseases into two main categories. One, and I will pretty much emphasize infectious diseases in this talk. Infectious diseases in the wild, unfortunately, are a little understood. There's very little knowledge of many of the natural diseases, and for that reason, it's difficult to understand what diseases that we see in captivity, what the infectivity and transmissibility of those diseases are for the animals in natural habitats, because we don't have a large understanding of their natural diseases to begin with. As far as captive diseases, and I again mean diseases that emerge or arise in settings where these animals are removed from their from their wild sources and then established in a zoo or wildlife facility, then leading to diseases that might be related to new adaptations of organisms because of the exposures to the other species. For example, at the National Zoo, we watched a particular parasite, a spiroid parasite from the slow loris, adapt itself to calotrichids, which are the marmosets and tamarins, via the cockroach. The cockroach, of course, which is quite a difficult, one of the vermin to deal with in a zoo, is the intermediate host for this parasite, and also relished by these marmosets and tamarins. So this parasite was nicely introduced over a number of years in a zoo setting. Management-related problems, which include such things as diet, environment, the various things in their background that could lead to diseases that they ordinarily wouldn't acquire in their natural habitats. And again, many of these have been worked out by trial and error, and in most cases effective ways of dealing with them have been established. And of course, there's one important aspect of the pathogen itself, and that is the host range. Diseases, for example, rabies has a very wide host range, and for that reason can affect many different taxa of mammals, whereas some other viruses, like herpes viruses, are often more host-specific, and there's little tendency of, with some exceptions, of that sort of virus to cross over into other species. So there are a number of factors that are important here to determine how diseases actually spread. Well, I just wanted to point out that we've come a long way in zoos as far as understanding some of their needs in captivity, particularly nutritional needs, which without good nutrition can lead to more susceptibility to disease problems. I mentioned that I'm going to emphasize infectious disease potential, and you can see that viruses probably have the greatest potential for catastrophic events, and again working both ways, from captive populations to wild and the reverse. Bacterial infections are often associated with stress and other underlying diseases. Fungal diseases are often more sporadic, and then parasites are limited by their life cycle. For example, if there's an intermediate host that's not available in the captive setting, why the parasite probably isn't a problem. On the other hand, I will illustrate several parasites that can be major sort of factors limiting the ability to propagate certain species because of a parasite that's either adapting itself to a new animal species, as I mentioned, or is infecting the animal as an aberrant host. I'd like to illustrate some of the disease categories that I've mentioned, beginning with viral diseases. The first disease I'd like to illustrate, rabies virus, portrays the transmission of a virus from a wild population into a captive setting. And this centered around the great epizootic of raccoon rabies that occurred from 1980 and is still in existence, traveling up the northeast, practically up into Canada. It began around 1980, and we have a facility in Front Royal, Virginia, in which there are many raccoons in the natural setting. And there was at that time a die-off in the raccoons that turned out to be rabies. And this was the beginning of the epizootic, which the National Zoo experienced cases 2 or 3 years later. You can see in this time chart the first cases occurred at Front Royal, Virginia, which is about 80 miles west of the zoo, and over the next 2 years traveled to Washington, D.C., where we had our first rabid raccoon in October of '82, and then the first case in a red panda associated with an encounter with a rabid raccoon. We had vaccinated, as we were aware of this disease, many of the susceptible animals, but in some cases the vaccine either didn't work because it's not proven in these species, or they weren't vaccinated at the right time. This is one of the outside exhibits, and the red pandas, which are actually somewhat related to the raccoon family, live in the outdoor exhibits and stay out all night, and apparently rabid raccoons would come in and encounter the red pandas and transmit the disease. We had at least 3 cases, and while raccoon rabies is considered the non-furious form in raccoons, it is just the opposite. It's these red pandas with rabies are very aggressive, and you can see here one of the red pandas exhibiting this aggressive behavior associated with the rabies. This was proven as a raccoon strain. Here you can see a section of the brain with very nice negri bodies, the intracytoplasmic viral inclusion that's classic of the rabies virus, but by using monoclonal antibodies, these strains isolated from the red pandas turned out to be the raccoon strain. During that time, we initiated a trapping program to trap raccoons to assess the rabies problem, and we're able to do some work in the raccoon populations in D.C., which of course are quite urbanized and plentiful, and had been the source of a number of cases. For example, at 1 month in 1983, we had 13 rabid animals found in the park. Right now, the disease over the years has become enzootic, and we see fewer cases. Well, we actually, in cases of raccoons that were rabid, we harvested brains and worked with CDC to continue to study this strain of virus. We found some interesting made some other interesting observations. For example, this is a salivary ganglion with a very nice inclusion body, negri body, which fits the pathogenesis of the virus as it comes through the salivary gland, and most likely making its way from that ganglion. This is an area map depicting the epizootic, which was initiated by raccoon hunters from the southeast U.S. Rabies has been enzootic in southeastern U.S., and the transportation of some of these raccoons by raccoon hunter clubs further north was believed to be the initiating factors that led to the spread. This slide is actually about 2 years old, and now the disease has actually crossed into upper New York State and into Canada. All of our staff and all of the susceptible animals in the collection are vaccinated routinely, and it's a disease now that is considerably under control, but occasionally we've a rabid raccoon surfaces. The next disease I wanted to talk about was a disease that has many implications in both wild and captive settings, and has been shown to be transmitted in both directions. I'd like to begin with the transmission of canine distemper from wild species. Again, in the zoo setting, it's mostly, we believe, from raccoons, in which this disease, canine distemper morbillivirus, is enzootic as well. The susceptible families of carnivores, as you can see, include the Canidae, Procyonidae, which includes the red panda and the giant panda, mustelids, viperids, the hyenas, and now the felids, which I will make some comments on the extension of the host range of this virus, of the canine distemper virus specifically, and morbilliviruses more generally. The signs of canine distemper in exotic species have some variation amongst the species. Conjunctivitis is a very common sign in practically all species, but For example, clinical signs may differ. In foxes, CNS can be aggressive like rabies, and in raccoons it can also be aggressive as opposed to rabies. In these three species, fox, ferrets, and raccoons, jaundice is often seen in animals with canine distemper, as well as some of the skin lesions, the classic hard pad disease, which I will illustrate. In other cases, the animals are found dead with very little in the way of any clinical signs. Here's a classic case of a raccoon with severe conjunctivitis that is associated with canine distemper. Now a red panda, a species that is exquisitely sensitive to not only street virus, but to acquiring the disease by using canine-derived vaccines, which I will talk more about in a few minutes. This is a European mink with conjunctivitis that one of 4 that developed canine distemper associated also with a modified live vaccine. An example of the classic hard pad or hyperkeratosis associated with canine distemper virus. And then the main changes are very similar, again, as they are in dogs, which include pneumonia, encephalitis, lymphoid depletion, and inclusion bodies. The inclusion bodies are fairly classic. This happens to be the bronchial epithelium from a red panda with vaccine-induced distemper, and you can see the nice eosinophilic intracytoplasmic inclusion body typical of morbillivirus infections. Also syncytial giant cell in the form of giant cell pneumonia, which is common. There's as I mentioned, a wide range of exotic carnivores that are highly susceptible, including the black-footed ferret, which was almost devastated by this virus, and represents now the reversal, the source of the virus, which practically knocked out those few remaining black-footed ferrets in their natural habitat, was thought to be from domestic dogs. And the other example of canine distemper virus, or a virus being transmitted to wild populations, is a devastating outbreak of canine distemper in the Serengeti National Park in Tanzania, Africa, in 1994. These slides depicting the lion outbreak were provided to me by Dr. Melody Relke Parker, who was the veterinarian in charge of the park at the time, and had the opportunity to work with this outbreak. The outbreak actually killed about 1/3 of the population in that park, which amounted to about 1,000 animals lost, and the source of the virus was attributed to the many unvaccinated dogs in the surrounding villages, in which distemper was proven, and perhaps was then carried into the lion populations by the smaller wild carnivores, including the spotted hyenas, jackals and other carnivores that the distemper was found in also. This lion actually is in the process of having a grand mal seizure, which was one of the classic signs in these populations. As I mentioned, they were worked up very intensively. Virus was isolated, and in fact was canine distemper virus, not some other morbili virus, and turned out to be closely related to dog virus, and also a raccoon, a virus isolated from a raccoon that transmitted this disease, caused the disease in an outbreak of big cats in California several years before that. Here we see one of the staff with Dr. Melody Relke doing a necropsy and working up the cases. It was well worked up with through the efforts of many investigators, including Dr. Appel from the Baker Institute at Cornell, who showed very nice correlations in tissues with immunohistochemical studies. As I mentioned, vaccine-induced distemper is a rather large issue in captive species, as well as now even those wild species that are now the host range seems to have extended to. And the problem being there's really no vaccine. There has been no vaccine that's safe for these exotic species whose adaptation to the existing vaccine products for dogs often lends the animal towards getting the disease. These are the species in which vaccine-induced distemper has occurred. You can see the red panda, as I mentioned, is one of the most sensitive, but also African wild dogs, black-footed ferrets, Maine wolves, gray fox, the bush dog, kinkajous, and fennec fox. There have been all documented cases of their contracting canine distemper from modified live vaccines, mainly from canine origin, that is, vaccines that are attenuated in canine cells. Now, with some work, there have been some vaccines that either have been identified as working. For example, there's an inactivated experimental vaccine that has been prepared by Dr. Appel that has, with some limited use, been protective in some of these exotic species. An attenuated vaccine that's prepared in non-canine origin cells, now called Galaxy D, is useful for some canids mainly. And then the new vaccine, which is now on the market, which is a recombinant vaccine, canary pox vectored canine distemper, a product of Roan Mario, or now called Marial, shows a great promise for a multi-species vaccine that is not infectious and can produce perhaps immunity without the hazards of causing distemper-induced disease. One of the things that limits, of course, testing these vaccines is the numbers of species we have to deal with. Sometimes there are not enough. We're dealing often with conserved species, endangered species, and of course virulent challenge is not possible. So most of the trials are done on the basis of serological conversion, and the hopes that these are comparable to what connotes immunity in the domestic species. the Maine wolf, and of course, as I mentioned, the most susceptible are the young pups and red panda pups, all very, very susceptible to canine distemper, for which we're hoping to obtain vaccines for. Now what I'd like to do is to talk about a very interesting virus that, if you remember my list of how diseases get into the various zoo settings, we mentioned food products. The baculovirus traditionally has been recognized as a virus of insects mainly, and more recently there's been virus strains mainly from Asia that have been infectious for the Penaeid shrimp, a virus called the baculovirus causing a white spot disease. Unfortunately, this virus was recently introduced into the U.S. in farm shrimp and produces a lethal virus called the white spot, which decimates populations of shrimp being produced for the shrimp industry. And the new observation in our setting was that we had a similar disease in crayfish, which it hadn't been reported before, that were purchased from vendors to feed our cephalopods, mainly octopus, cuttlefish, and nautilus, which thrive on the crayfish. Well, in 1995 we had an outbreak in the feed crayfish. Now these are crayfish that are purchased from vendors who obtain these from different sources, usually from the south or southeast. It's not always clear as to where they come from, but these crayfish actually, when we buy them in large lots, we were having die-offs and actually did necropsies and found that they had all of the characteristics of what appears to be a white-spotted disease of shrimp. Here you can see the exoskeletons here are modeled. You can see these white sort of target-looking changes in the shell constituents, which are really due to the virus in the epithelium underlying the exoskeleton, which produces the shell and forms irregularities. Here you see these dark purple structures which represent intranuclear inclusion bodies, and when you do electron microscopy, these are the characteristic. They're rather large viruses, greater than 200 nanometers, and form these interesting arrays in the nucleus. This is an example of DNA hybridization using actually reagents for the Penaeid white-spotted shrimp disease, indicating it's basically the same virus. There's perfect homology between the two viruses through the amplification techniques and DNA hybridization. So it represents a new disease that really hasn't been reported in wild crayfish, and the status of the shrimp in Penaeid or in the U.S. now is that there has been some evidence that it is in the wild populations of some of these invertebrates, but so far we haven't proven that fact in the crayfish. Well, what I'd like to do is to continue on with some of these concepts of disease movements and some of the concerns and implications. One of the things that zoos are doing today are providing ways of replenishing the populations that are being reduced in nature for various reasons. And some of the implications that go along with that include disease monitoring. That is, assuring that if captive populations are going to be reintroduced to sustain the natural populations, they must be disease-free and not reintroduce particularly new diseases. Diseases, as I mentioned, in captivity can be often different, and wild populations may be naive to these diseases, and so we don't want to then bring in new diseases, nor subject those reintroduced animals to perhaps diseases that occur in the wild that they may be naive to. So these are very important concepts when you're moving animals back and forth between captive and natural habitats. And I'd like to illustrate it with the possibilities of a catastrophic virus getting into wild populations. We've had a lot of success propagating a particular endangered species called the golden lion tamarin, which is indigenous to the Atlantic rainforest in Brazil, and have produced successfully a number of generations that then would be destined to go back to Brazil and several preserves that we have there. And during that time, we had a number of diseases emerge in captive settings, but one that began in the 80s as epizootics at about 10 zoos of what appeared to be a fatal viral hepatitis. Histologically, it appeared like akin to some of the viral hepatitis syndromes in other primates and in humans. It had an acute onset with lethargy. Seizures seemed to be part of it, elevated enzymes and bilirubin, very rapid course with death and few survivors. And as we looked into this initially, we were not able to identify what particular agent it was. We did transmit it to obtain more material so that we could study this virus from natural case using liver from an infected animal into several common marmosets and produce the same syndrome. And as you can see here, this is the open body cavity of one of these common marmosets, showing enlarged, pale, somewhat mottled liver and peritoneal hemorrhages, also not shown but common are peritoneal effusions, and also some in the pericardial sac. Well, it turned out that this agent was identified as an arenavirus, closely related to, if not the same as lymphocytic choriomeningitis virus, which causes a flu-like illness in humans. And in fact, two of the veterinary caretakers actually developed titers but no illness to LCM, and suggesting, or again implying now, not only a possibly catastrophic virus, but one that was zoonotic. And it turned out that the source was from a practice that was common of feeding neonatal mice to these marmosets and tamarins. Now, it occurred in a variety of callitrichid species, so it's not limited to the golden lion tamarins, but there was a high incidence in the golden lion tamarins. And we, in fact, were able to isolate LCM from mice from a particular outbreak, which we then which were fed mice at the time, and we then related that virus back to the agent that we recovered from earlier cases. So it was clearly LCM that was causing the hepatitis. Here you see a section of liver with a disarray of the hepatic plates. You can see that there are some necrotic cells, scattered inflammatory cells, and this hyaline structure, which is typical of the disease, that characterizes the disease in this form of hepatitis. We developed an immunohistochemical procedure which stains liver cells with viral antigens using antibody from a guinea pig strain of the LCM, and so we can actually make diagnose the disease from formalin-fixed material. Here are the culprits, the neonatal mice that have been fed and were determined to be the source. Of course, now this recommendation has been made. The recommendation has been made not to feed any mouse parts or products or neonates whatsoever. And in fact, now over the years, this has led to an abeyance of the disease. Occasionally, we see cases still that are related to wild mice. We've shown that this disease also has occurred in wild mouse populations in some zoos, but it's of a considerably low incidence. Pygmy marmoset, which is also very susceptible to the disease and has shown very also high incidences. Well, these animals were never returned to the wild with this virus because we were able to sort it out, and before we sent any back, all these animals are screened, and any that are even seropositive, even though that probably connotes conferred immunity, are withheld from being reintroduced, because this virus also has some latent characteristics. These tamarins are actually allowed to have free access to an area in the zoo so that they can learn to fend for themselves before they're sent into the wild, so sort of a boot camp-like situation. And with the surveillance, not only for this virus but for many of the other primate diseases, we've been very successful in returning these, and there's been now over 150 that have been reintroduced with several generations of births now in Brazil of this species, now living in the wild, but still monitored under surveillance. I'd like to move on to another virus, and in this case it represents an emerging virus or a novel virus that also provides disease concern for zoo settings. It doesn't happen often, but over periods of years there have been a number of viruses that have emerged that are infectious for exotic species. For example, the morbilliviruses, as I've mentioned, have extended their host range now to not only terrestrial carnivores, but to species like cetaceans and pinnipeds, producing outbreaks of fatal disease globally. More recently, morbilliviruses have been attributed to deaths in horses and humans in Australia, and that represents sort of a new viral, emerging viral disease. In our setting, the virus I would like to talk about is a new herpes virus infection. It's an endothelial-tropic virus that occurred in our young Asian elephant Kumari that we bred and were raising at the National Zoo. She was doing quite well up until 16 months of old, when she developed a rather vague disease characterized by just anorexia, lethargy, some colicky signs, and depression. And the disease had a rather short course, which lasted about 5 days, and she died with a terminal cyanosis of the tongue unnoted. At necropsy, you can see that she was really quite in good nutritional condition and had some changes that I'll summarize as oral and intestinal ulcers, disseminated petechial hemorrhages over serous membranes, and pericardial effusions. So I mentioned the tongue seemed to have a very characteristic cyanotic appearance to it. Here you see the around the oral cavity. This is the trunk up here, then the lower jaw with the these punched out ulcers around the mouth. Quite severe endocardial hemorrhages. Also petechial hemorrhages over, this is the spleen with capsular involvement, which also were noted over all of the intestinal and the parietal peritoneal linings of the abdominal cavity. The hallmarks of this disease histologically are the presence of intranuclear inclusion body in the endothelial cells, in the heart, the liver, the tongue, and some other areas. You can see that there's, along with this injury to the endothelial cell, extravasation of red cells and edema, and hence the pathogenesis appears to be that of quite severe, as the virus replicates, quite severe capillary injury with hemorrhage and edema and death and cardiac arrest. Those intranuclear inclusion bodies by electron microscopy showed the presence of viral structures that were consistent with herpes virus morphology. You can see again now the inclusion body with the within the endothelial cell, the center structure being the inclusion body, and at higher magnification you can see both empty and nucleocapsids with the characteristics of herpes virions. The virus was pinpointed as a herpes virus by amplification techniques using consensus primer, which is a gene for the DNA packing process in the cell, and it fits the subfamily of beta herpes viruses, although there's no relationship to any of the known human or herpes viruses that we're familiar with. The other important fact is that we were able to retrospectively identify 6 other cases of this herpes virus from the SSP stud book records that were available, and after searching, we found cases that match the case definition of this disease in Kumari, and we're able to obtain slides and material to then show that by light microscopy and EM, and also by PCR technique, that these animals were all infected with this same, what appears to be a new endothelial tropic virus. The just to go back on this slide for a second, you can see the animals were mainly in North America. Actually, they were all originated in North America at various zoos in the United States and Canada. The animals were usually young, ranging from 16 months to 26 years. That was the oldest, but mostly were young calves with an even sex distribution, a course of very short length, and a death very rapidly after the onset of the disease. We don't know really the source of this virus. We expect it might be an indigenous elephant virus. There has been one more case recently, but that occurred in an African elephant, again from a zoo, being hand-raised, with the virus not exactly a replica of the Asian virus, but very similar to it in its structure. This is a representative, what represents the various groups of subfamilies of viruses, and you can see the elephant herpes virus appears by molecular information to fit into the beta viruses, but this is not yet conclusive. It might be sort of somewhere between beta and gamma of viruses. I'd like to continue now with some other interesting pathogens to show you how these make their way into zoo settings. Beginning with a disease caused by Clostridium botulinum, It's a disease that's well known in wild waterfowl and causes rather huge mortalities at different times of the year, often associated with environmental factors and the migratory patterns of the birds. In waterfowl collections, which are small and in smaller settings, that is, like in zoos, we also experience similar outbreaks from time to time. It's much like a microcosm of really what's happening in the wild setting. Here you see a lineup of waterfowl that died in an outbreak of botulism at the National Zoo. As you know, it's a disease that causes paralysis of the neck muscles, so that the birds, in effect, if they're in the water, basically lose their ability to hold their heads out of the water and can drown. At necropsy, the findings are usually of birds that are in good nutritional condition. There may not have been much evidence of having eaten anything recently, but aside from that, there are virtually no morphologic changes that can be attributed to the botulism. The source of the disease in these small settings usually are the carcass of an animal, usually another bird that has been struck with fly maggots, which then allows the botulism organism to proliferate and then makes its way into the water habitat where the birds are. In our case, we have a series of connected ponds in which, after a fairly dry autumn, a carcass apparently from one of the waterfowl species that died then was washed into the pond by a sudden storm one evening. We actually found the carcass, as you see here. That was actually retrieved from the pond and was considered to be the source of the maggots, which once they make their way in the water are eaten by the birds and then affected by the toxin that's building up. Proof of this is often by obtaining serum from the birds that are still alive and inoculating mice, one of which is protected with a botulism toxoid. And in this case, we were able to clinically show that that was the case, and also were able to isolate botulism toxin from the maggots, which were sent to the wildlife disease lab in Madison, Wisconsin. So this represents, again, another sort of type of disease that we see in both wild settings and captive settings, showing similar features. The next disease I'd like to discuss is an example of a bacterial disease carried by one of the 3 R's, as I mentioned before, rodents. Rodents, rats and mice, are often responsible for a number of diseases, the one in question being leptospirosis. This is a bush dog. It's a South American canid that appears to be very susceptible to leptospirosis, as we had these animals in an outside exhibit and were experiencing fatalities, and at NECROPSY we found a series of changes that pointed towards a disease like leptospirosis. Here you see the lungs with extensive hemorrhages throughout the surface and within the parenchyma. The other hallmarks are that of jaundice with a deep yellowing of the aorta at the bottom, and you can see also changes in the cortex with these white rays that represent necrosis associated with the organism. The diagnosis of leptospirosis is not always very easy. In the very acute cases, it's difficult to identify the organism, or there's not enough time to mount a serologic response. But in this case, we were able to actually trap rats from the area, and were able to find that they had titers to icterohemorrhagicae, and also found by using special stains, a Warthin-Starry stain, that their kidneys contain numerous organisms compatible with leptospira. So this is a disease that many warm-blooded species are very susceptible to. The canids, in fact, some of the marine mammals are, in addition, very susceptible to lepto. And are routinely vaccinated now. I'd like to mention several interesting parasites. As I mentioned earlier, that parasites in captive settings are often limited by their life cycle, such that in a direct life cycle they can be of some importance, and depending on what indirect life cycle they have, may or may not be problems in zoo settings. Balis ascaris is a roundworm that can be found in several different wildlife species. Procyanus is found in the raccoon, and there are several other species that occur, the skunk of which also contains Balis ascaris. Their larvae are often very infectious for other warm-blooded hosts, such that they don't complete patency, but migrate aberrantly, often through the CNS, and cause a considerable amount of damage, often leading to serious CNS deficits or death of the animal. I'd like to illustrate this particular parasite, most likely from the raccoon, in another species of lion tamarin called the golden-headed lion tamarin. It seems that they have a predilection for this a parasite, and it's not known whether there's a specific predilection or whether it's associated with the behavior of this animal, if they ground feed more or tend to eat things from the environment that other animals wouldn't. Well, here's the briefly the life cycle, which is fairly straightforward, at least in the raccoon. But the eggs, once they've been exposed to the outside and can be picked up by any of the small rodents and or even children are susceptible to this disease via the quite permissive ability of these parasites to migrate through these species. This is a disease now that is carried by wildlife and can affect both other wildlife species or get into captive settings and cause disease in animals like I'm illustrating here. The process is very difficult to diagnose ante-mortem. Specifically, clinical signs are usually those of CNS origin with ataxia and other motor deficits that lead to the animal's inability to locomote or to fly. And the reason is that these parasites actually migrate through the spinal cord and often get into the brain. Here you see cross sections of the typical appearance of Balis ascaris procyanus or any of the other species in this golden-headed lion tamarin. The way this happens is that feces from, in this case with the golden-headed lion tamarins, was deposited in an area where they had access to it, which relates to the management practices of building exhibits so that this can't happen. These exhibits were from a smaller zoo that had the outside exhibit so that raccoons could actually get to the top of the exhibit and defecate. The raccoons have a way of defecating in exposed areas like tree limbs and anywhere where there's opportunities for the feces then to get into a position where these other animals can be exposed to it. Well, these are examples of other exotic species that have been afflicted by the larval migrans that takes place. So, represents a macaw from a group of these citizen birds that were put out on an island where there were mulberry trees, and during the mulberry season the raccoons ate the mulberries and then defecated, and their feces actually turned out to be this really bright sort of purple fecal matter that appeared to be quite attractive to the macaws. Hence they ingested it, and then months later developed the visceral larval migrans, eventuating in having to have the group euthanized. So again, some of these are, this is an example of having this knowledge so that those developing the exhibits can be aware of such diseases and make arrangements to prevent by building the exhibits properly so that raccoons or other animals can't get near them or or have direct exposure to the animals that are being kept in the exhibits. A woodchuck, which is and also squirrels, which are common victims of this very aggressive raccoon or skunk parasite. The other parasite I wanted to mention was a similar situation, but one that represents a parasite that's has a very symbiotic relationship with its indigenous host, but in the aberrant host, the parasite can be lethal. And this is Paraelaphostrongylis tenuis. It's indigenous in whitetail deer, and it's with the increasing populations of white-tailed deer in the east, along the East Atlantic coast. This parasite has caused problems not only in wild cervids that are not sympatric with white-tailed deer, because this parasite is lethal for them, but also for private collections of ungulates and in particular llamas. We have well here this shows the natural host being the white-tailed deer, and the intermediate host being several species of snails and slugs, so-called terrestrial gastropods. The aberrant host, as I mentioned, include other cervids, and in nature include various species of cervids, including moose, wapiti, red deer, caribou, and reindeer are highly susceptible, and others. The aberrant host includes some domestic species, sheep, goats, and some exotic species that are usually raised in captivity. In our situation, involved sable antelope, 2 species of oryx, including the Arabian and the scimitar horned oryx, and at least tentatively bongo antelope. The parasite has an incredibly complex life cycle. beginning with the third stage larva, which is produced by the intermediate host, and is either eaten by the white-tailed deer, who completes the life cycle over a period of several months by ingesting either the whole snail or the larva itself. The larva then migrates through the stomachs into the spinal cord and eventually deposits itself going through a fourth stage to the adult, which actually lives on the meninges and then become fertile, and eggs are laid, migrate back through the venous sinuses in the meninges back into the lung, coughed up, and the first stage larvae are swallowed and then go out in the feces and then picked up by the land gastropods and repeat the cycle. Now in a setting where there are other hosts, the aberrant host, you can see how then these are transmitted to the aberrant host merely by either picking up the third stage larvae or inadvertently taking in one of the gastropods. Now, the National Zoological Park has a 3,000-acre facility in Front Royal, Virginia. Here you can see in the foothills of the Shenandoah Valley. It's a beautiful setting where we've had opportunities to raise and propagate endangered species, and to do both physiological and reproductive research for the benefit of these conserved species. Here are the buildings and the site where the animals are housed. The whole facility does have a large perimeter fence that keeps out some of the white-tailed deer, but occasionally white-tailed deer can get in, but also the intermediate host, of course, can travel from the outside perimeters into the paddocks where these ungulates live and carry the parasites to them. Here's a scimitar horned oryx, which again is one of the susceptible hosts. Clinical signs in the well, as I mentioned, this parasite has a symbiotic relationship with white-tailed deer and rarely, if ever, cause any clinical signs. But in the aberrant host, the common signs are those of ataxia or what appears to be shifting lameness, which eventuates into paresis with head tilt circling and then often total paralysis. There's really no good way of making the diagnosis of this parasite ante-mortem, and it requires a post-mortem examination and finding the worm tracks in the worm itself in the spinal cord. Here's cross sections of spinal cord from sable antelope, and you can see these lesions in the one side of the cord are attributed to the migration of the parasite. Occasionally, but very uncommonly, you'll find fourth stage larvae. Here you can see, and this is it depicts the spinal cord, and you can see the white arrow actually pointing to a fourth stage larva of one of these parasites in the spinal cord of a sable antelope. But more than likely, one requires to exhaustively go through many sections of usually they're found in the upper spinal cord and posterior part of the brain. And you can see these cross sections of nematodes with virtually no reaction around, indicating that they're actively migrating, are diagnostic. In some cases you don't find cross sections of the parasite, but you can see hemorrhagic tracts, and in cases where there is some chronicity, you can often see inflammatory remnants of the parasite, which has moved on in the form of lymphocytes, plasma cells, often around vessels, and occasionally eosinophils. Well, controlling this parasite again is not easy. There are some indications that some of the newer parasiticides like ivermectin have some effect, but it's difficult to incorporate a program like this with the variety of different species that we have, and the various problems that go along with that. So another approach is to try to reduce the deer and the ability of the parasites to make their way into the zoo. And so we have actually attempted to improve the fencing and to provide gastropod barriers, and really also use the resistant species, which usually include the equids and some of the other deer species. So we've had virtually no problems with this disease with L's deer or paired David's deer. So they seem to be resistant, and those are the ones that are put into the areas where we have the higher incidences of this parasite. The barrier method is virtually removing as much foliage from the perimeter fence, and to produce barriers with a gravel on both sides of the fence, which prevents the parasites from crossing over, the gastropods, the snails and slugs, which do not like to move on rough surfaces. They prefer grassy surfaces, and so that reduces the ones that are exposed to the deer on the outside from getting in. And this actually, when it was begun several years ago, seemed to reduce the incidence, but it's expensive and it has to be redone on a regular basis. And at one point, when it wasn't kept up as well, we did notice an increase in the incidence. So again, those are examples of a number of diseases. In summary, I just wanted to point out how important it is after you've seen a number of these diseases operate. I think that I've been to be able to develop some of these ideas about the dynamics of the diseases, how they move back and forth. We don't always know all of the details. We don't know, for example, we know less about diseases moving from the captive populations to the wild populations, but clearly it's important for zoo the zoo planners and leadership to be aware of these disease problems. As you can see, the implications are that exhibits, if built incorrectly or at that level, or if animals are moved without good disease monitoring, why diseases can be then spread in both ways. And